Okay, hi, how's everybody doing tonight? Tonight we're going to continue our discussion about osmosis and passive transport. And before we continue it, I just want to go back and review what we discussed yesterday a little bit on passive transport, and this is what we had in our notes. Let's get this out of the way. We said passive transport is a very important concept in biology because it's the way we explain how molecules, in particular small molecules, can move into and out of a cell and cross that selectively permeable membrane called the cell membrane. And we said, and we placed in our notes, that there are a few different forms of this passive transport. There's simple diffusion, where molecules generally just move down their concentration gradient, no energy required to do that, recall, and they move from areas of high concentration to low concentration through the cell membrane, maybe even through the lipid layers. And oxygen and carbon dioxide are good examples here of simple diffusion. They're so small, they have no charge on them, those molecules, they're able to go through the lipid bilayer. Then there's a type of passive transport called facilitated diffusion, and that's also molecules are still moving down the gradient from high concentration to low concentration, but they're using the protein channels to help move the molecules. Maybe the molecules are too big to get through the lipid layer, or they're charged molecules and they won't be able to pass through the region of the lipids where it's hydrophobic and hates water. And so they'll use the protein channels as a way to facilitate their way in and out of cells. But it's also no energy required. That's the main thing with passive transport, where molecules move but no energy is used by the cell to do it. And today I'm going to talk a little bit more about osmosis. We said that's a specific type of diffusion or passive transport where molecules are still, again, moving from high concentration to low concentration, but this is the movement specifically of water molecules. Okay, so these are the notes that you have, and again, we're going to fill in as we go along. That's your homework assignment, to fill in the notes, and then we'll discuss this a little bit further tomorrow and do some labs that will apply what we're talking about. So before we move on and talk about osmosis, we have to talk about what a solution is. Solutions are usually combinations of two or more compounds or substances mixed together. Usually one might be water, and in the diagram you have here, you maybe can write it a little clearer. You have sugar inside this beaker of water, and when you stir it around, the sugar gets sort of diluted and mixed around completely in the water. And so solutions are basically a homogeneous mixture, a mixture that's uniformly distributed of two or more substances. Sugar water is a good example of a homogeneous mixture when we stir up or dissolve the sugar in water, or even salt water is a good example of a solution where salt is diluted inside some water. And so we can write down some examples as well, and we'll use the sugar as an example since our picture says sugar in it. Now, whenever we talk about solutions, there's always going to be something doing the dissolving and something that gets dissolved. So what we call the substances getting dissolved, like in this case it's the sugar, we call those substances the solute. And this molecules or the compounds that do the dissolving, well, we call those things the solvent. So in this example, we have the water is the solvent because it's dissolving the sugar, and the sugar would be the solute because that's the stuff getting dissolved. And if it was salt water, then it might be the salt that was the solute, and again, the water would be the solvent. Water is called the universal solvent because it has the ability to dissolve many things in biology, and so we kind of nickname it the universal solvent. But those are two important terms that we need to discuss when we talk about solutions and osmosis. And fill in your notes with these important terms. You can pause the video if you need to. Now, let's look at some situations that a cell might be placed in. So, and we're going to call these the important keywords. Osmosis, we said, is the movement of water across a cell membrane. But there are a couple important terms that I'd like us to learn. And let's look at this cell. This cell is being represented here by yellow. And here's the cell membrane, this thin line around it. And they're showing arrows, and the arrows are representing which way water would flow. So let's look at this diagram a little bit closer. And the dark molecules that sort of black here in your diagram, they might be gray, and they're the smaller ones. Well, those represent free water molecules, and the bigger molecules are pink in the video here, and they might be just slightly shaded in your notes. Well, those are going to be the solute molecules. It could be salt, it could be sugar, it really doesn't matter. And inside the cell, well, we have the same molecules. We have water, and we have these dissolved solutes in pink here. Now, if we look outside the cell, we can see that there's certainly a lot of solute, a lot of these larger molecules compared to what's inside the cell. Inside the cell, there's a few of them, but there's certainly a lot more outside the cell. And if we stay in analyzing the outside solution of this cell, we can see that there's very little water. When you have a lot of solute or a lot of sugar, they're going to take up the majority of the space in the solution, and you'll have less water in your solution. And if we compare the water outside the cell in these dark, smaller circles, we see there's a lot less compared to inside the cell. And the arrows are representing the way water would flow. We know by passive transport and osmosis, water moves from an area of high concentration to an area of low concentration. No energy needed. It's a small molecule. It can move from high to low down the concentration gradient until it reaches equilibrium. And that's exactly what would happen if a cell was placed in this sort of environment. This environment is referred to as a hypertonic solution. And simply a hypertonic solution refers to what's going on the outside of the cell here. There is a higher concentration of dissolved solute or dissolved substances outside this cell. Very little water outside the cell. And when the cell was placed in this hypertonic environment or hypertonic solution, we can see the water would rush out of the cell. Now, this might affect the cell. If it was a plant cell, it might all of a sudden become sort of shriveled up. If it was an animal cell, that cell might also lose its water and become shriveled up as well. So it's an important keyword we have to understand called a hypertonic environment or hypertonic solution, and it just refers to the solute, in this case, on the outside of the cell being at such a high concentration, much higher than inside the cell. And if there's such a high concentration of solute outside the cell, then there's going to be very little water, and that's represented again by those small circles. There's very little of it outside the cell as compared to inside. So water will rush out of the cell as indicated by the arrows, and the cell will shrivel up. So that's one way that cells can be affected by unbalance or an imbalance in water molecules between its inside cytoplasm and outside environment. Let's look at another situation and fill in your notes for hypertonic. Now, this picture shows a very different environment. This picture shows the solute concentration outside the cell being pretty low compared to inside the cell. If we look at all the larger round circles inside your notes, is a lot more of them inside the cell. I'll show you with my cursor here, as compared to outside the cell. It's the complete opposite of what we saw before. Now, there's a lot of water outside the cell and very little water inside the cell. Now, this type of situation is referred to as a hypotonic solution. Again, the cell has been placed in an environment that's different from its cytoplasm. And in this case, there's very little water inside the cell. There's a lot of solute, could be sugar or salt, doesn't really matter, but there's a lot of those molecules inside the cell cytoplasm, and if there's a lot of solute, there's very little water as compared to outside the cell, which has a little solute but a lot of water. And as the arrows show, the water would flow into this cell. Osmosis would try to reach equilibrium, and the water molecules would move from where they're highly concentrated outside the cell and start to move into the cell until they reached equilibrium. That's referred to as a hypotonic environment. The cell in this picture has been placed in a hypotonic solution or hypotonic environment where there's a lot of water outside the cell, very little solute outside the cell. That's why there's so much water. And that water is going to rush right into the cell and could swell it up or even burst it if there's too much diffusion of water into the cell. So this is a hypotonic type of solution. The cell is again placed in it, and there's very little solute in this solution outside the cell, but certainly in a hypotonic environment, when there's very little solute, they'll make up a lot of water molecules inside the solution itself. So let's okay, so let's go back and let's make sure you have the notes on that. A hypotonic environment, when cells are placed in that, water will rush into them because there's a lot more water outside them than inside them. And the last example I would like to bring out, and we'll put in our notes, is called an isotonic solution. And in an isotonic solution, the environment outside the cell and inside the cell is just almost identical. If we look here, it looks like there's equal amounts of solute inside the cell as compared to outside the cell. It almost seems as if it's reached equilibrium, the dissolved substances, whether it be sugar or salt. And because there's equal amounts of sugar or salt or solute inside the cell and outside the cell, there's also going to be equal amounts of water inside the cell and outside the cell. And we can see that the cell has basically reached this state of equilibrium, dynamic equilibrium. So the arrows are showing the movement of molecules would not only continue, but some of the water might move into the cell, some of it might move out of the cell, because molecules, remember, are always in motion. And so the movement of molecules wouldn't stop even when they've reached equilibrium, but there would be no net gain or net loss of water molecules in this sort of environment if the cell was placed in it, because all the solute and water is equal. And so we refer to this as an isotonic solution. A solution that has equal concentrations of solute, dissolved substances, and water as another solution. So we have three different situations in which a cell can be placed. Once again, you have hypertonic, you have hypotonic, and you have isotonic. And you should play this video over again and make sure you understand what these terms are. They could be confusing. Now, tomorrow or the next day, we'll do a little demo where I'm going to take a potato and I'm going to place them into different sort of beakers with different concentrations of water. And so this is not on your notes, but we'll do this demo together and we'll see how placing a potato in different concentrations, different from what its cells are. Its cells are normally 90% water and 10% minerals or solute. And if I take it and I place it in a beaker that it's very different than its own cellular contents, like potato beaker 1, I'm going to put it in a salt water solution. And in beaker 2, we'll place it in a solution that's very similar, almost identical to the potato cell's internal cellular environment. And then in the last beaker, we'll place it in a distilled water. So we're changing the environment that the potato cells are being placed in. And we'll sort of try to predict what will happen to the cells. Will they swell? Will they shrivel? Or will there be no net gain or loss in terms of water? And we'll hopefully see osmosis in action. Now, the last few things on your notes is to predict what will happen. So what I want you to do is, I've written down and I've given you this information. An animal cell is typically 95% water and 5% solutes. You know, there could be some salt and minerals in with the cytoplasm of an animal cell, but it's mostly water. And I want you to predict, if you place this animal cell in this first beaker, and I wrote that down, that the water was 85% water and 15% solute. Okay, what would happen to the cell? Would it shrivel up? What would be the change in the cell? You can write it right underneath there. I left some room for you. Would the cell shrivel up or would it swell? Remember, osmosis is the movement of water from high concentration to low concentration. So think the animal cell has 95% water. We've placed it in an environment that has 85% water. Right underneath here, write your prediction or your change in cell. Will the cell lose water or gain water? The same thing for the next one, but the next beaker is 100% pure water or distilled water. We've placed the same animal cell now in a different environment. And the third beaker has the animal cell going into an environment that's exactly identical to its cytoplasm, an isotonic environment. So predict underneath the change in the cell. You can write it out. You can draw with arrows which way water will flow, but predict what will happen to the water and which direction will it move. And I'd like you to do one more. The next page on our notes shows a red blood cell. It also contains 95% water and 5% other ions and materials and minerals, things like that. And I want you to predict again what would happen if I took this red blood cell. Let's make another one. And let's put it in a beaker. And this beaker has 100% water. Pure water, no solute at all. In fact, I'll put 0 over here to represent that there is no solute. It's distilled water. And then I'll put the red blood cell in another beaker over here, and it's 10% salt solution. So could you predict how much water is in the solution? If it's 10% salt, how much water? Well, if you guessed 90%, you would be right, because every solution has to add up to 100. So there we have a solution that I'll place the cell in with 10% salt and 90% water. And in this one, I'll put the red blood cell in an environment that's identical again to its cytoplasm. It has 5% salt, and again that would mean that it would be 95% water. And I would like you to predict what will happen to the red blood cell. Will it swell? Will it shrivel up? Will it stay the same? And you can write your prediction right underneath the beakers here. And then we could stop. There's one more example. We'll discuss it in class with the fish, which way will water flow in a fish tank if you have freshwater fish versus salt water. But we'll discuss that in class tomorrow. But what I'd like you to do is predict the red blood cell in various concentrations and the animal cells put in various concentrations of solutions. And remember, it's all about osmosis and hypertonic, isotonic, and hypotonic environments. And I hope this was helpful, and we'll go over it more tomorrow in class. Well, have a nice evening. Bye-bye.